Welcome back, friends. In today's video, I am going to discuss on the uh, male and female reproductive organs in Cycas plant. So in my last video, I had already mentioned that the Cycas, the reproduction of Cycas, it takes place by means of both vegetative and sexual method. In vegetative reproduction, we have it takes place by means of the adventitious buds, or what we call bulbils. Whereas in sexual reproduction, we have seen that the cycas is strictly a dioecious gland. That means both the male and the female organs, they are produced on separate plants. That is, the male flowers, the Microsporophylls, and the female flowers, the Megasporophylls, they are formed on different cycas plants. So here in today's video, I am going to discuss on the structure of both the Microsporophylls as well as the Megasporophylls. So let's get started. So here the male cone. Now the male cone of Cycas, it develops singly on the apex of the male plant, or sometimes we find a few, 2 or 3 male cones are growing at the apex of the stem in other Cycas plant species, but in most of the Cycas plant species, these male cones, they develop singly at the growing apex of the stem. This male cone is a compact structure. As you can see here, it is a very compact structure, cylindrical or ovoid in form, and also it is woody in texture. And also each cone is very large, up to about 50 centimeter in length or more. In some Cycas species, it can reach the length of up to 50 centimeter or even more than 50 centimeter. Now here in the case of Cycas plant, the apex of the shoot of Cycas plant, this shoot apex here is utilized in the development of the male cone, so that the apical growth of the stem itself is stopped. Okay, the apical growth is already stopped. Why? Because it is used in the development of this male cone. So here later on, when a new lateral bud develops, a new lateral bud will develop at the base of the peduncle. This lateral bud will start to grow straight upward, and it will push the male cone to one side. It will push this male cone to one side. Thus here we see that the male plant, it shows a sympodial branching. Okay, the male plant shows a sympodial branching. Sympodial branching, it occurs when the terminal bud ceases to grow. Okay, usually because of this terminal cone has formed, and an axillary bud, a new lateral bud will develop, and it acts as a new leader shoot. This is another picture showing a male cone in Cycas. This is quite short as compared to the first one. You see here the different Cycas species, they showed different, they have different different sizes of male cones. Okay, the sizes of the male cones, they differ in different plant species. This one also is another Cycas plant species, and this is the compact structure forming the male cone, and these are the Microsporophylls. One more thing here, we see that the Microsporophylls, they are arranged in the form of a what we say a spiral. Okay, in the form of a spiral, where the youngest microsporophyll, they are formed at the top, and the oldest Microsporophylls are found at the lower part of the cone. Here you can see these are the Microsporophylls. Okay, now each cone, it consists of a central axis on which these Microsporophylls, they are compactly and spirally arranged. As I have said in the earlier, that the youngest Microsporophylls are formed at the top and the oldest at the bottom. So this way of arrangement of these Microsporophylls, that means they are arranged in acropetal succession. Acropetal succession meaning the youngest at the top and the oldest at the base. Okay, this is, I had already explained that when this male cone, it usually arises singly, and later on when a new apical bud grows, it will push the male cone to one side, and new foliage leaves will start to grow, and later on a new male cone will form here. Okay, see, even here we can see that this already pushes this male cone to one side. These are the new foliage leaves, new crown of foliage leaves, and this is the newly formed male cone. Now here, this male cone, if you take a look at the LS section, longitudinal section of a male cone, what you will observe that, see, these are the Microsporophylls. Many Microsporophylls can be seen, and this part in the middle is called the cone axis. So each cone, you will find the male cone axis in the middle with the Microsporophylls arranged in a compact structure, and they are spirally arranged in acropetal succession. Now each Microsporophyll, now if we talk about only this single Microsporophyll here, you will see that, okay, this one, if we take out this single Microsporophyll here, this Microsporophyll, it measures about 3 to 5 centimeter long and also 12 to 13 millimeter wide. And here you can also see it is flattened, wedged shaped woody structure. Also, if you observe one more thing here, that the Microsporophyll, what you will see here, that it has two surfaces. Okay, the adaxial surface or the upper surface, and the abaxial surface, that is the lower surface. Even here in the LS section, you can see they have the Microsporophyll, they have the upper surface and the lower surface, upper surface and lower surface. Okay, so the what are these which you will find in the lower surface? They are nothing but they are the Microsporangia. Okay, the sac-like structures which contain the microspores, which carry the microspores, in which the microspores form. They form inside these microsporangia. Now, okay, we will take a look at this single microsporophyll, and we will take a look at its lower surface. Okay, this is the lower surface, the abaxial part. Now, if you take a look at the abaxial part, the basal narrow part here is very sterile. Okay, the basal narrow part is sterile, and this flat distal position is fertile. Now, beyond this fertile part, what you will see here is there is an upper extended fertile part. Okay, sorry, this upper extended part here is also sterile, and it is called as the apophysis. Okay, this is what we call the apophysis, and it is also sterile. Now, this fertile part, as I said, it contains many microsporangia. Okay, these are the microsporangia. Internally, okay, this microsporophyll, if you take a look, if you take the TS section of this microsporophyll, what we will observe internally that the microsporophylls, it consists of parenchymatous ground tissue in which a large number of mucilage duct, not only mucilage duct but also the vascular bundles are scattered. Okay, the mucilage duct are scattered and the vascular bundles are arranged in rows. So this is the transverse section of the microsporophyll, and you can see here these are the microsporangia with the microspores inside. Now, if we take a look at these microsporangia, here there is a distinct median sterile ridge on this lower abaxial surface of the microsporophyll. Okay, each microsporophyll, it bears a large number of microsporangia, and these microsporangia, they are borne on either side of the ridge. One more thing here, if you see, is that the microsporangia, they are either scattered or they are formed in groups of 3 to 6. Okay, sometimes 3 or 4 or 6 microsporangia you will find in groups. Now, these microsporangia in groups, here you see here many microsporangia, they are formed in groups. This is what we call a sorus. Okay, a group of microsporangia is called a sorus. And also you see here, intermixed with the sorus, intermixed with these sorus, what you will find, the hairs. Okay, you see here the hairs. These hairs, they are called the sorul hairs. And these sorul hairs, their function is to help in the dispersal of the microspores which are formed inside the microsporangia. They help in the dispersal of the microspores, and also they protect the young sporangia. Now, the microsporangia, if you take a look only at a single microsporangia here, these microsporangia, they are unilocular, meaning they are single chambered, okay, unilocular, oval, and also sac-like structures attached with a short stalk. Okay, they are shortly stalked and they are attached with a stalk to the microsporophyll. Now, the microsporangium encloses a large number of microspores or the pollen grains inside. Okay, each of these microsporangia, all these microsporangia, they encloses a large number of microspores inside. So next we will take a look at the development of each microsporangium, okay, and also the formation of the microspores. Now each microsporangium, here we will talk about only the single microsporangium, how it develops. So the microsporangium, it develops from a single cell, single sporangial microsporangial initial. Okay, these are the microsporangial initials. Now the microsporangial initial cells, they arise from the hypodermis. Okay, these microsporangial initial cells, they arise from the hypodermis of the sporophyll. Now the group of the initial cell is called an archisporium. Okay, a group of these initial cells, they are referred to as the archisporium. Now these archisporium, the cells of the archisporium, it will divide by periclinal division to form an outer primary wall cell and an inner primary sporogenous cell. Periclinal division meaning division which is parallel to the tissue, to the cells. Okay, so you see here, these will form the outer primary wall cells, whereas these are the inner, these two are the inner primary sporogenous cells. Now these outer cells, the primary wall cells, they will again divide repeatedly by periclinal as well as anticlinal divisions to form 3 to 6 layered wall of the microsporangium beneath the epidermis. Okay, you see here, this one. These are the outer primary wall cells which has already divided and give rise to 3 to 6 layered wall of microsporangium beneath the epidermis. Now these other cells, that is the primary sporogenous cell, will also divide repeatedly. Okay, they will divide repeatedly to form a mass of sporogenous tissue. The outer primary wall cell will form a 3 to 6 layered wall of microsporangium, and also the primary sporogenous tissue will also divide repeatedly to form a mass of sporogenous tissue. Now here, if we take a look at the sporogenous tissue, the outermost layer of the sporogenous tissue, it will form the diptum. Okay, the outer sporogenous tissue, it will form the diptum, That is, the tapetum is a specialized layer of the nutritive cells, okay, which will provide nourishment to the cell. Now, what are these cells inside? Some of the sporogenous tissue will become enlarged or will increase in size, and they will become the mother cells, okay, the spore mother cells, microspore mother cells. They will enlarge and become microspore mother cells. Now, but other cells, not all of these will become microspore mother cells. Some will become they will get disintegrated with the tapetum, okay, they will get disintegrated with the tapetum, and they will provide nourishment for these microspore mother cells. So again, I will repeat that the sporogenous tissue, the outer wall of the sporogenous tissue will form the tapetum, and the other cells, they will increase in size. Some of the cell, they will increase in size and become the microspore mother cells, and other cells, they will disintegrate with the tapetum so as to provide nourishment to the growing microspore mother cells. Okay, now in the next step, these microspore mother cell, they will start to divide meiotically, okay, divide meiotically to form 4 haploid microspores. You see here, after they divide here in this step, they form the spore tetrads. These are the tetrads which will finally form the microspores or the pollen grains, the haploid microspores. So, if you take a look at the structure of the Microsporangia, it become boat shaped. Okay, it become boat shaped. And also one more thing here, a mature Microsporangia is sessile. Okay, here I had already mentioned that the Microsporangium, they are shortly stalked. But when the Microsporangium matured, they are either sessile, that is without a stalk, or they are shortly stalked. And also they consist of 5 to 6 wall, okay, 5 to 6 wall layers. Now these 5 to 6 wall layers, the outer single layer, that is the exothecium, and the innermost layer here, we know that it is the tapetum, and the rest are the endothecial layers. And also the wall of the sporangium, here it encloses a large number of these haploid microspores. Now here the tapetum, it does not remain as it is. This tapetum is used up during the development of these microspores. So in a mature Microsporangia, only the signs of the disintegrated tapetum can be seen. Okay, only the disintegrated tapetum can be seen in case of a mature Microsporangia, because it is already used up for nourishment during the development of these microspores. Now what happened here, you just take a look at this diagram. At maturity, what happened, this central axis, it will elongate so that these compact microsporophylls, which are arranged compactly, they will become separated. And as the microsporophylls, these single microsporophylls, they become dry and separate from each other, the microsporangia also starts drying. Okay, so each of these microsporangium, after it becomes dry, each microsporangium dehisces by a longitudinal slit at its anterior end. Okay, there is a longitudinal slit at the anterior end of each microsporangia in order to release these microspores to the outside environment. But here the microspores, they start to germinate even before its liberation from the microsporangium. Okay, remember this one important point, that the microspores, they start germination before its liberation from the microsporangium. So that's it for the explanation on the male cone. In my next video, I am going to explain on the female cone of Cycas. Thank you.